Okay, so in this last video, we're going to change gears and start talking at translation by looking at the genetic code. So remember, in the central dogma, we have transcription, which converts DNA into mRNA. That mRNA in eukaryotic cells leaves the nucleus and is translated into protein. And so the genetic code specifies how a sequence of nucleotides codes for a sequence of amino acids. And so scientists were trying to think of how a sequence of nucleotides, 4 nucleotides, could code for 20 different amino acids. And here's all of the amino acids. We talked about these on day 2 of the quarter. And scientists were starting to think about this, and mathematically, they thought it made the most sense to start with a triplet code. So they needed a code to code for these 20 different amino acids, and there's 4 different bases. And so if the code was just 2 letters, that would only code for 6 amino acids. So you'd have a 1 in 4 chance of an A, T, C, or G, and another 1 in 4 chance of A, C, T, or G. And so you just multiply those together to get the number 16. So that didn't make sense because we needed to reach 20 amino acids. And so this led Francis Crick to hypothesize that we needed a 3-letter code, because that would give us 4 times 4 times 4, right? At those 3 different positions, there's a 1 in 4 chance of ending up with a different base. And this could give us 64 amino acids. So that covered the 20 known amino acids and suggested that there might be some redundancy in this genetic code. So this led to a number of studies by Crick and Brenner, who showed that the reading frame, so how these codes were read, could be destroyed by adding or subtracting 1 or 2 bases. So they focused in their experiments on this R2 gene of a T4 bacteriophage. A bacteriophage is a virus that infects bacteria. And they used this chemical carcinogen called acridine. And they knew that acridine created point mutations by either adding or subtracting only 1 nucleotide. Okay, so that's where we'll start. So I'm not going to use the amino acid code, but I'm going to essentially use our reading frame. So to think of these amino acid or these nucleotide codes as words. Okay, so we're going to start with this sentence: the fat cat sat. Okay. So when they treated with acridine and they deleted 1 letter of this code, the protein that they were studying being made from this bacteriophage was no longer the same. And so in this case, when you delete that A, then this sentence, all of a sudden you have the, and then this sentence basically becomes gibberish. Similarly, if you added 1 letter, so instead of deleting this A, and of course you can see here the reading frame shifted backwards, which made it gibberish. Here we're adding an A, and so the reading frame is being shifted forward, right? So now that A is here, which means the TC, which finished the word fat, is now beginning this new word. And so we still end up with gibberish, and the proteins were not functional. But then they did these really smart experiments where they crossed over different mutant viruses, and this mixed the mutations. And so now the sequence was either plus or minus 2 or 3 nucleotides. And so when it was plus or minus 2 nucleotides, the sentences would still be gibberish. But if 3 nucleotides in this case were added, we had a reading frame back intact. And so this would be an example of another 3-letter word being inserted here. So the 1 fat cat sat. So we're back in the reading frame. So these experiments, by adding or subtracting 1 to 2 nucleotides and ending up with something not functional, but then if you could get to 3 nucleotides, showed Crick and Brenner and confirmed that these 3 bases specify a particular amino acid. And so we refer to these 3 bases as codons, okay? So a reading frame, right, is how we read the codons, and they're read 3 at a time. And so that's what set scientists to figure out what exactly the code was. So 2 scientists, Nierenberg and Mathieu, developed this way to synthesize RNA. So this was a real breakthrough. And they could synthesize it. And the first RNA that they synthesized was this polymer of uracils. And they added this to a test tube with ribosomes and the machinery you needed to make proteins. And what they got out was a polymer of phenylalanine. And so their conclusion was that the triplet code UUU is the code for phenylalanine. And this was all very exciting. So they went on to make AAA, CCC, and GGG and figure out what amino acids they coded for. And then they had a breakthrough and figured out how to mix sequences. And they found all of how each triplet code encoded for a particular amino acid. But they also found one start codon, so that was AUG, and it coded for methionine. And they found several stop codons, which caused the polypeptide to stop growing. And so here's the code that they figured out with these really bright experiments. And so if you have a code that's UUU, as I said, that will give you phenylalanine. But there's some redundancy because UUC will also give us phenylalanine. Another thing that you can see, and we'll come back to this later, is that a lot of codes, right, a lot of codons, right, are similar in the first 2 letters, so U and C, right? And then it doesn't matter if you have a U, C, A, or G in that final position, you'll end up with serine. And this is true for proline and leucine. Here is our start codon, so AUG codes for methionine. And then we have a few stop codons, so UAA, UAG, and UGA. You are not going to be asked to memorize this list, but you should be able to, if given this information, right, you should be able to convert a triplet code into an amino acid, or if given an amino acid, be able to figure out the triplet code if given the appropriate information. So a couple more things about the genetic code. So the genetic code has some redundancy, right? So all but 2 amino acids are encoded by more than one codon. So let's go back and look at this, right? So we have methionine, that's our start codon. It's only encoded by one codon. But most of them, right, have several codons that then can be converted into an amino acid. They're also unambiguous. And what that means is that one codon never codes for more than one amino acid. So when we get a codon, we know exactly what amino acid that codon is going to code for. They're non-overlapping, so codons are read one at a time. They're nearly universal, so all codons specify the same amino acids in all organisms with a few minor exceptions. And they're conservative, and I pointed this out. So what that means is that if several codons specify the same amino acid, the 2 first bases are usually identical. Okay, and we'll practice in class with what exactly these terms mean. Okay, so knowing the genetic code and the central dogma means that biologists can predict the amino acid sequence by looking at a particular DNA sequence. So here, for example, we take our template strand of DNA, right, and it's TAC. So that means that our mRNA is going to be AUG, okay? And that AUG, because we know we've cracked the genetic code, we know that's a methionine that's going to start the protein. And we can do this, right, going 3 letters at a time to figure out the mRNA and then how that's translated as an amino acid. And so you can pause that video and go back to the code and try, or open up your textbook and try to complete this yourself, although we're going to practice it in class as well. You can also determine mRNA and DNA sequences that could code for a particular sequence of amino acids. But remember that because of the redundancy, there could be more than one DNA sequence that could code for the same amino acid sequence. So now that we've talked about the genetic code, we're going to talk about what are the consequences of mutations in this code. And by mutation, what I mean is any permanent change in an organism's DNA. This is going to modify the cell's information archive. It's going to change its genotype, and it's going to introduce new alleles. And there are different types of mutations. The 2 we're going to talk about are point mutations that result from one or a small number of base changes, or chromosome level mutations that are on a lot larger scale. And so a common way to introduce a mutation is during replication, right? So recall that DNA polymerase, right, it has proofreading ability, but it's not 100%. And so here we have our original DNA, and in S phase, right, we have, we're replicating this DNA. So this bottom strand here in red represents the replicated DNA. And here we have a base pair mismatch that wasn't caught by DNA polymerase, even though this forms a different shape. And so that mismatch, right, these are separated into daughter cells. But here, that mismatch, right, is now a mutation, right? It's going to get copied into the new daughter cell. And so that's how a point mutation could get introduced. Okay. So let's talk about types of point mutations. So point mutations are categorized by the effect they have on the amino acid sequence. Okay. So we're going to consider this example here where we have a DNA sequence, TAT, TGG. Okay, I'm just going to focus for now on these 2 first codons. So this will code here for UAU, UGG, which will give us a tyrosine and a tryptophan. Okay, so this is our original polypeptide. But here in this instance, let's imagine that we have a point mutation. So we had a problem during DNA replication, and we had a base pair mismatch, and this introduced a C instead of a T in the DNA. Okay, so that C means that we're going to have now, and I think this should be a G, right, in that next position, right? And so this, right, is going to still, this codon is going to still give us tyrosine. So even though the amino acid sequence has changed, right, it didn't change, the codon didn't change the amino acid that was made, right? This amino acid would still be tyrosine. And so there's no change in the phenotype, and it's always neutral with respect to fitness. So that would be a silent mutation. A missense mutation is a change in the nucleotide sequence that changes the amino acid that's specified by the codon. So here let's focus on this next codon, and let's imagine that thymine was introduced here, which would mean that the mRNA would have a uracil. So instead of having tryptophan being produced by the codon UGG, we now have UGU, and this encodes for the amino acid cysteine. So this is actually going to change the primary structure of the protein, as well as perhaps the secondary and tertiary structures. And it's important to point out that this can be beneficial, but it could also be neutral or deleterious, right? So it really depends on what part of the protein that this is in and how it changes the conformation of that protein and its function. A nonsense mutation is a mutation that results in an early stop codon. So this is easy to look at. Here we've introduced an A into the DNA sequence. That should give us, I don't know why the textbook has the wrong letters here, a uracil here that would introduce a stop codon. And this would lead to, if this was this is an actual stop codon, this would lead to mRNA breakdown or a shortened polypeptide. And this is usually deleterious, right? Because you're just stopping the protein from being made midway. The last one is a frame shift mutation. Okay, so again, silent means we're changing the code but not changing the amino acid. Missense, right, means that we're changing the amino acid. Nonsense is a stop codon. Frame shift is switching our reading frame. This can be caused by the addition or deletion of an amino acid. So here we've actually inserted a new amino acid, right? So this would be like those T4 bacteriophage experiments. And so we've inserted amino acid here, so our whole frame has been shifted over, right? So we have more nucleotides here at the end. And in this instance, right, we end up with now a serine, and everything after it has also been shifted. So this is the reading frame being shifted, and it alters the meaning of all subsequent codons, and it's almost always deleterious. So mutations can have different impacts on fitness. They can actually be beneficial and increase the fitness of an organism, its ability to survive or reproduce. Mutations can be neutral and have no effect on the organism's fitness. This is especially true if the amino acids are similar, so polar to polar, non-polar to non-polar, or they can be deleterious and decrease the fitness of an organism. Most point mutations are either neutral or deleterious, and some mutations are not in the coding region, but can still affect gene expression, perhaps if they're in the promoter region or part of the operon, right? So this is just something to think about. It doesn't actually have to be in the protein to have an impact on an organism's fitness. So the last thing we'll talk about are chromosome mutations, and I just want to explain different types of chromosome mutations. So here's a normal chromosome, and I'm showing this with different genes along this chromosome here in different colors, different banded colors. And here you can see that in this chromosome, a big chunk of this chromosome with both genes A and B has been deleted. So a segment of the chromosome is lost. This will shorten the overall length of the chromosome, so these can be identified fairly easily in a karyotype. An inversion is where a part of the chromosome breaks off, flips around, so the B will go over the A, and then rejoins. Okay, so you have this opposite position here, and this can produce problems, especially during crossovers, right? Because you have this flipping, but also problems with gene regulation because the promoters don't always come in the same place. We can also end up with duplication events. So here A and B have been duplicated, and so now they're AB, AB, so we have twice as many of these genes around. We also have translocation. So in translocation, a section of the chromosome breaks off and becomes attached to another chromosome. So here, B, right, has been broken off. Sorry, this blue one has been, or D and E, sorry, have been broken off and combined here, and then B and C have broken off and then combined with this gray chromosome. So it's pretty typical in cancer cells. The genome of a cancer cell is very unstable, and so they accrue a lot of point mutations, but also chromosomal mutations. And so it's pretty typical to see a karyotype with a lot of aneuploidy. So here, for example, is chromosome 19, right? These should have 2 copies because we're diploid. And you can see that this chromosome 19, there's 6 homologous chromosomes for chromosome 19. So that's a very typical, that's an aneuploidy. You can end up with translocations. So here, chromosome 1, it's picked up part of one of these other chromosomes here. I don't know if it's chromosome 8 that's orange. You can end up with deletions that will shorten the chromosomes. So for example, chromosome 17 over here, this is a lot shorter than it should be. And you can you can get duplication. So here, chromosome 2, you can see a duplication event because of that chromosome's much longer. So that's it that I have for this video. Again, just go over the concepts of kind of analyzing the genetic code and be able, before you're coming to class, if given the right information, to convert the code to an amino acid or an amino acid to the code. So that's it for this video, and I'll see everyone in class. Thanks, bye.